The overall goal of this procedure is to describe a comprehensive system for sequencing challenging cancer specimens that combines wet lab reagents, standardized controls, and a standalone bioinformatics suite.
This method can help answer key questions in the cancer field, such as how to sequence low quantity and low quality tumor biopsies to achieve accurate DNA variant analyses and interpretations. The main advantage of this method is that samples that are difficult to sequence, or those that simply have limiting DNA quantities available, can be quickly and reliably analyzed using single source reagents and controls and fully integrated bioinformatics software.
Generally, individuals new to this method will struggle because of the complexity of targeted NGS, which this method simplifies. However, the procedures for library quantification and sample pooling require special attention to achieve uniform coverage across all sample libraries.
This protocol integrates wet and dry bench processes for the sequencing of challenging cancer specimens. The first step is quantification and qualification of DNA to determine the number of DNA template copies that can be amplified by real-time PCR. Begin the procedure for sample quantification and quality control by preparing a sufficient amount of a master mix in a microcentrifuge tube. Use the following volumes per sample: 5 microliters of 2x master mix, 0.5 microliters of primer probe mix, 0.5 microliters of inhibition primer probe mix, 0.05 microliters of rocks, and 2.95 microliters of diluent. Add 9 microliters of the master mix into each well of a 96 well plate.